AI stepping into the scene is important because a single genome is just so much information for researchers. A single human could spend their whole lifetime trying to understand even just like one part of a genome. So by training these models on so many genomes from plants, humans, animals, bacteria, viruses, we've been able to scale up an understanding over so much of life that was quite difficult to get at previously.
Welcome to Talk Bio to Me, the show where we sit down with the people building the future of biology. I'm your host, Flo Lumsden, founder of Chorus Studios, a podcast production company where we transform complex technologies and breakthrough ideas into human stories that educate, entertain, and inspire. This year, we headed to SynBioBeta, the world's largest conference for synthetic biology.
Increasingly excited about AI actually making an impact in the lab.
Shaping the future of biotech. From breakthrough therapies to AI that's beginning to design biology, I discovered one of the most exciting technological revolutions of our time. Okay, talk bio to me. Welcome to Talk Bio to Me.
It's great to be here.
We're so excited to have you and to learn about your work. To get us started, would you just give a brief introduction what your name is and what you're working on?
Yeah, so I'm Samuel King. I'm a PhD candidate at Stanford. I research bioengineering broadly. I currently work on using and developing AI models for better understanding biology, but also designing new biology. So we do this at the very molecular level, designing new DNA sequences and proteins and all of the machinery that composes us. And we've now recently learned through lots of developments that these AI models are very good at understanding this level of information about human biology and the rest of biology.
Physics, does that play in? And on the molecular level, I would think there'd be molecular physics. Is the AI really good at physics too?
So the models that we work on, not necessarily. We kind of hope that they, in a way, capture the physics about DNA sequences just implicitly through the language of DNA. You can definitely create these more like physics-informed models, but the ones that we work with are very much like ChatGPT. But instead of it being trained on text from the internet, it's just trained on genomes, like millions and millions of genomes. And through seeing all of that language of DNA, the model starts to pick up these principles and rules about how DNA has been designed by nature. From there, since the models are generative, just like how ChatGPT is, you can use it to then generate new sequences that are coherent and capture the functional properties that you want.
So is this based on an existing LLM or is this a custom one?
So there's some of the architecture has been borrowed, like transformers is really popular, but the models that I've worked with have a slightly newer architecture that's better at handling the really long range signals that you want to capture in DNA sequences, because if you think about these really, really long sequences like genomes, there's these cool facts where if you disentangle the whole genome or something, it stretches really far. But in English language or other text, the signals that you want to capture are a little bit more local. Like within the same sentence, you can generally understand what is being talked about. But in a genome, you could have a gene somewhere over here that's millions of DNA base pairs away from what it might be important for or something. And so we need better architectures for AI models that are able to capture these really long range signals.
Are you working on the AI for it or are you using the existing tools you have to do the research?
Yeah, so my PhD work so far has mostly been on applying these models. I am trying to also dabble in developing the models as well. So recently what we were able to accomplish was we used these models to design an entire genome that actually works in real life. And so this was the first ever demonstration that you can use AI to generate a coherent whole functional genome. And the way we approached this was we generated genomes of bacteriophages, which are viruses that infect bacteria. The idea was we wanted to, of course, explore the capabilities of the models to reason about like a whole genome, and if it does that well enough, can it create an entirely new one on its own? But then also for bacteriophages specifically, they're quite useful as potential therapeutics against like antibiotic-resistant pathogens and things like that.
Oh,
Yeah.
cool. So you developed a new genome for a new virus that can infect bacteria
Yeah.
and help with diseases that are resistant to antibiotics that are bacterial.
That's the hope for the
That's the-
future. There is a lot of rising concern that antibiotic resistance will become a leading cause of death in a couple decades. And also currently the way we use antibiotics, it's quite,
Fast and loose.
yeah, you just like, it just like kind of destroys your whole like microbiome or like lots of your microbiome. And it's very like a lot of off-target effects. And we want to go towards a future where we can create much more precision medicines. And bacteriophages, naturally, they have a very specific propensity for their target host. And so you can maybe someday start programming bacteriophages to go somewhere in the body and specifically target the one singular pathogen that's problematic.
Right, like you would take a sample from someone who's sick from a bacterial infection and use that sample to create a custom antidote.
Exactly. That's kind of how it is currently done. So there are phage therapies, bacteriophage therapies that are already in use for these kinds of cases. But maybe someday we'll be able to do the design well enough where a lot of it can just be simulated and there'll have to be less invasiveness of needing to sample from the patient and things like that.
Okay, so backing up a bit, you created a new virus, but the extrapolation that
Adrian.
Adrian
Yeah.
was saying that it could one day we could maybe use AI to compute new biologies for like a new animal or something.
Yeah, I mean, we're headed towards a pretty crazy sci-fi future where biology will be end-to-end programmable, and we might be able to, we're getting to the point where we're hopefully understanding genomes well enough that we can start creating designer genomes with specific applications. And AI stepping into the scene is important because a single genome is just so much information for researchers. A single human could spend their whole lifetime trying to understand even just like one part of a genome.
I'm going to ask you a really basic question real quick.
Yes.
And I should have just looked this up before between the panel and now. But the difference between a genome and DNA, DNA makes the genome.
Yeah, cellular organisms all have their genomes. They're like heritable instructions encoded as DNA. But we see other types of genomes, like viral genomes, can be encoded by like RNA.
Okay, wow.
And different formats of DNA and RNA.
Okay, yeah. Okay, cool. I think I maybe understand a little.
Yeah, sorry, I should have made that clear.
No,
Like,
it's like everybody here knows that except me. So I'm just like, yeah, I've heard that. I've heard of a genome. I know more clearly in my mind what DNA is than a genome. A genome sounds like more information about the full animal or full species.
Yeah, you can think of it as the set of instructions, or I guess the list of parts that are needed to produce life, and it's what's passed down from parent to offspring. Yeah.
Cool. Where were we? Oh, we're talking about creating animals that don't exist. I interrupted you to ask you what a genome was.
Yeah, I was talking about AI stepping in. So by training these models on so many genomes across like all domains of life, so we take genomes from plants, humans, animals, bacteria, viruses, and we train the model on all of it. And it learns these kind of principles across all of these genomes of what makes up a gene, or the component for like expressing a gene, or even like smaller things. Like the model has a pretty good understanding of when a single mutation can be detrimental or non-detrimental to a genome. And in this way, now we've been able to scale up an understanding over so much of life that was sort of quite difficult to get at previously. And in this field, like we've been able to get quite far in understanding and engineering genomes, but now we're hoping to then scale it up even further using these models to reason about all of these long scale things in genomes better for us.
when I think about like what makes a life successful on the planet Earth, like it needs to be able to do all these processes of, collecting vitamin D and breathing somehow. So I know the genome creation, I guess the ideation has to fit within the world that we live in. Can the AI like extrapolate like, okay, all of these animals have these things in common, so this is necessary to live on this planet?
We do see that the models have started to get an understanding of the essential and non-essential components that are required for life. And we can now maybe leverage that information to start building out like synthetic cells or something,
Yeah.
or minimal genomes that you can then use as like your scaffold to start adding all of the other things that are extraneous, but
Right.
code the functions
Would be nice to have. Yeah. Not
Yeah,
like give it that basic survivable function
exactly. Yeah.
and add on
Yeah.
features. Yeah, that makes sense.
And from the other angle, we've also in biology learned quite well like what a lot of the essential components are. But it's nice to put some reliability into these models now to just ask it, okay, I want this genome that has this property and function. Can you write me something that captures everything I need?
Yeah.
Instead of having to spend like all those painstaking hours piecing everything together yourself as a biologist.
Right. Yeah. And I'm sure it also just because I use it for my podcasting to give me ideas, like blog ideas from the episode or title ideas and things that wouldn't have thought of. It's just great to like get more ideas that then you wouldn't have that you would have on your own.
Yeah, most people would argue that these models are synthesizing like new ideas by essentially just combining knowledge and information that it's seen. And that, maybe we'll soon see like true creativity from these models. But there's a lot of merit actually to this ability of like cross-pollinating and, especially in creating new DNA sequences. Like you might want to borrow this gene from this organism and put it in the context of this genome and add the right mutations in other places that make sure that works. And the model seems to be able to do that. And that kind of counts as a form of creativity, I think.
Yeah, totally. What about like designer babies? Do you see that coming?
I think that future is also on the horizon.
Or at least less ill babies. That would be good.
Yeah, we want to see less diseases upon birth. Gene editing, I think, will have a role to play in that. There's some increasing interest in gene editing, like embryos, I think. But when people hear designer babies, it sounds like very cosmetic.
Yeah, sounds very cosmetic.
But hopefully we're implementing these technologies to eradicate disease as opposed to leading towards like problems like eugenics and things like that.
I'd slightly said that to be a little controversial.
Yeah.
I don't really mean it. Well, is there anything else you want to talk about? Oh, I could ask you what you're enjoying about SynBioBeta so far, or why you decided to come this year.
Yeah, I mean, SynBioBeta has been an amazing experience so far. It's actually my first time here. It's great to get all of these brilliant minds in one place. And I feel like there's been so much serendipity. I just run into somebody that I maybe sort of knew of by name or like have heard of or never met before. And all of these like new ideas and interesting conversations have been happening. And I think creating an environment for serendipity is an amazing thing to do for researchers and people working in industry. And SynBioBeta is perfect for that. Yeah.
Great answer. Is there anything that you've learned about that was particularly exciting or inspiring to you, or just, cool?
There's been lots of things. I think I'm not as exposed to the world of like drug development and pharma. And I've been hearing lots of talks around how to accelerate like timelines or paths to clinic and developing drugs. And I think that's been quite insightful for me. And it's really nice to like step out of my typical bubble of what I do research in and hear all of these other perspectives, especially from like industry leaders, because I'm still a PhD student and I'm pretty embedded in like an academic environment. So hearing from all of these people who have startups and other companies, it's been pretty cool.
Can kind of validate what you've been thinking or maybe challenge some of your assumptions about the industry.
Yeah, definitely. Yeah.
Okay, if we have time, one last question about advice for someone who's considering the PhD route.
Yeah. A PhD is I think people can have misconceptions over what a PhD really is. I've always viewed it as a empty canvas where you have an opportunity to get to decide the specific questions or problems you want to solve. And doing a PhD creates an infrastructure for that. You're surrounded by lots of brilliant people and provided resources by an institution. And having that like creative freedom over things you're passionate about, doing a PhD is one of the best places you can pursue something like that. And so I think for anybody who wants to do a PhD, they should have that kind of spark of like something that they're really quite curious about or a problem that they really want to solve in the world. I think the other way to think about it as well is that a PhD isn't a narrow thing. I think a lot of people say that they avoid doing a PhD because they think that it will narrow them into like one extremely niche field of science or something. But PhDs, especially nowadays, are incredibly interdisciplinary. And honestly, every single day in my PhD, I felt like I've been drowning in new information. And it's just been the most enriching exploratory experience. And I think it's opened my eyes to like so many different things about the world. And so I would view a PhD as anything but narrow. And I hope that people can see that about PhDs.
Oh, I love that. Even I did think it was a narrow route. So you've opened my eyes. Maybe I'll go create a blank canvas to explore some part of the universe I'm deeply curious about.
Yeah, it's such a unique type of thing that I think many more people should really consider heavily doing. Yeah.
That's great. Great advice. So lovely to speak with you.
Yeah, thanks for having me.
Thank you for coming, Samuel. And we'll be in touch. I'll be following your research.
Great. Yeah, thank you.
Don't forget to subscribe or follow Talk Bio to Me on Spotify, Apple Podcasts, YouTube today for episodes dropping very soon.